Good morning, everyone. Thank you very much for the invitation to PSBMB annual convention. I'm Kozo Watanabe from Ehime University, Japan. I'm a full professor of Molecular Ecology and Health Laboratory at Center for Marine Environmental Studies at the university. Today I will talk two topics. One is stream macroinvertebrate biodiversity assessment, and one is dengue virus surveillance of vector mosquito in the Philippines. Before the lecture, let me briefly introduce about Ehime University, my university. And our location is here on the Shikoku Island. Then Japan has 49 prefectures, and each prefecture has at least one national university. And our university, Ehime University, is the national university in Ehime Prefecture. Then population size of our city, Matsuyama City in Ehime Prefecture, has about 500,000. Not so large as like Tokyo or Osaka, however not so super rural. Then this Shikoku Island is connected by 3 bridges to the main island. And our place is famous place for orange, and also known as a tourist spot, famous tourist spot in Japan. So if you have time, please visit our place in the future. The Molecular Ecology and Health Laboratory, this is my laboratory. We use molecular genetic tools like NGS in 2 areas. Freshwater ecology, we use insects and bacteria as study systems. For biodiversity assessment using metabarcoding or environmental DNA analysis, and also evolutionary analysis using bioinformatics and genomics tools. And also second area is health or eco-epidemiology area, vector-borne disease such as mosquito and dengue virus analysis, and antimicrobial resistance bacteria in water environment is also one of the foci of our group. Our group, this is country profile of group members: Philippines, Indonesia, Taiwan, Romania, Bangladesh, Mozambique, and Japan. Our group is the most internationalized group in the university. The Philippines, now we have 5 postdocs or PhD students. And in this March, 2 additional PhD students from the Philippines will join. And 1 plus 1 is my wife. She's from Pampanga. She's Filipina. And she's not an official member, however, she is taking care of Filipino and other international students in my group. And plus 2 is potential 2. This is opening 2 PhD positions. At the end of my lecture, I give small advertisement about this opening 2 positions. Then our group is also doing many overseas projects in the Philippines and Indonesia. Also, recently, our laboratory launched International Collaborative Research Laboratory at Laguna campus of De La Salle University. Here, we are now installing facility and equipment for molecular analysis. So I brought 2 topics, one freshwater biodiversity and second eco-epidemiology research topic. The first one is ecological influence of sediment bypass tunnel on macroinvertebrates in dam fragmented rivers by DNA metabarcoding. This was published in 2018 in Scientific Reports. First author is Jose Serrana, my PhD student. He graduated from De La Salle University. About sediment bypass tunnel. This is done, and sediment bypass tunnel transports sediment from upper to lower fragments of dam. Then this contributes to reduce sediment accumulation in reservoir and then maintain active storage capacity of the reservoir and lead to longer operating lifespan of reservoir. So this is this SBT is important from engineering perspective. This photo is outlet of SBT discharging a lot of sediment and water during flood event. And in this study, we focus on ecological perspective as an additive or additional effect of SBT. So from the SBT, sediment discharge, then this sediment contribute to recover riverbed condition of outlet river ridge. So the riverbed condition and macroenvironment committee at dam outlet recover with year after SBT operation. This is our hypothesis. Actually, no study has not tested this hypothesis. So our study was a world first hypothesis testing. Then in this project, we used amplicon-based next-generation sequencing analysis. We collected bulk macroinvertebrate bulk community samples, then homogenized and extracted DNA from community samples, and we use mitochondrial DNA CO1 marker, then amplified, then NGS, next, then got a huge amount of sequence information from the bulk community samples for metabarcoding analysis. So in this study, we aim to assess the influence of SBT operation on macroinvertebrate community in dam fragmented rivers in comparison to reference rivers via DNA metabarcoding analysis. So our study area was Swiss because SBT has been operated only in three countries in the world: Swiss, Taiwan, and Japan only. Therefore, we went to Switzerland in this project. There are 3 SBT dams were studied in the Swiss. Then these 3 SBT has different operation years. This SBT dam very old, 92 years old. And however, this SBT dam only operated only for 2 years. The sampling size and macroinvertebrate collection. So we collected samples from upper fragment and lower fragment before the SBT outlet and after. However, out of the 3 SBT dams, 1 dam outlet, SBT outlet was very close to the dam side, therefore we could not collect before SBT from this 1 SBT dam. Then in total, 8 study sites from SBT dam river. We also collected from dam fragmented rivers to rivers. These rivers had two only dams, but no SBT. Then free-flowing rivers. These rivers do not have dams. Then we collected from each river, we collected upstream and downstream sites. So in total, 8 reference sites were studied. Then in total, 6,853 larval individuals were collected from the 16 sites. And this is the result. After quality filtering, we got about 2 million sequence reads. Then meta-barcoding analysis, 16 from the 24 morpho families. Before DNA analysis, we morphologically identified at family levels. The 16 out of 24 were found. 28% of the reads and 29% of the morphologically identified specimen at family level. The right-hand side indicates total sample abundance of the 16 locations and total sequence abundance of each family. So each plot indicates family. For example, this one had high abundance based on morphological analysis and also showed very high sequence abundance from NGS analysis. They showed very good positive correlations. That means highly abundant morphological family were also detected many times by NGS. Then we recovered up to 131 species based on meta-barcoding from 36 families. And this meta-barcoding analysis, we use 150 base pair lengths of barcode. The most 46% of leads match to Vitis alpinus. This is this mayfly species, and this is widely distributing in the Alps and known as a very abundant species in this area. And this is a species composition of each location. Then this is the most important result. We evaluated beta diversity between upper and lower sites of river using Bray process dissimilarity index based on the meta-barcoding data. So red indicate dam fragmented rivers show, and green free-flowing rivers. Dam fragmented without SVT indicated higher community dissimilarity between upper and lower sites within river than free-flowing. That means probably these dams reduced migration event across the dams, or dams made different environmental or habitat conditions across the dams. Then different environmental conditions of the sites lead to higher community dissimilarity than free-flowing. Then dam fragmented rivers with SVT, out of the 3 SVT dams, 2 SVT dams showed relatively similar level of community dissimilarity to free-flowing. This means suggests SBT contributed to recover the habitat conditions and ecological conditions, as we expected. However, one SBT done, Solis, that showed similar or more higher level than dam fragmented river. That means in Solis dam, SBT did not recover the environment and ecological conditions. Then this plot indicates relationship between SBT operation years and community dissimilarity level. And they show negative correlation. That means old SBT dam tend to show lower dissimilarity. However, young SBT, like Solis dam, 2 years old, showed relatively higher dissimilarity. That suggests that certain length of or period of SBT operation year is necessary to show ecological positive influence. This is summary. We are able to use DNA meta barcoding data from collected macro-invertebrate rabbit to identify sample at the species level, estimate richness and diversity, and calculate community dissimilarity between upstream and downstream side of 7 alpine rivers. Then we report a positive influence of SBT operation on macro-invertebrate assemblies downstream of SBT outlet at 2 rivers. This symmetry value of the upstream and downstream assigned Solis dam were relatively similar to dam fragment size or even higher than the other sides. The dissimilarity between the macro-invertebrate community of the up and downstream side decrease with increasing SBT operation years. So let me move to the second topic, eco-epidemiology, surveillance of dengue virus in individual Aedes aegypti mosquito collected concurrently with suspected human cases in Tarlac City, Philippines. This paper has been just published recently, 2 weeks ago, in Parasite and Vectors. And this study was done by Jim Barinji, my former master's student from UP Diliman and Los Baños. Also, Dr. Tadeusz Carvajal from De La Salle University. He was my former PhD student and currently doing postdoc, JSPS postdoc at my research group. So dengue virus is that this transmission is occurring between circulating like this mosquito human. Then Aedes aegypti is the major carrier of the dengue virus in the Philippines. The dengue virus has 4 serotypes, and each serotype has many genotypes. Then one of the challenges of this study was individual-based dengue detection from single mosquito. Detecting dengue virus in field-collected mosquito is difficult and requires specific laboratory skills. Then most former studies rely on mosquito pool-based detection. They pooled around 10 to 50 individuals usually because it is cheaper, and also because of the small amount of dengue virus in a single mosquito. However, this pooled approach allows only to estimate minimal infection rate, like this, number of positive pools and total number of mosquito individuals analyzed. However, this estimation needs a big assumption, which is only one infected mosquito is present in a positive pool. So if one positive pool includes several positive individuals, we cannot count these multiple positive individuals in the estimation of minimum infection rate. Therefore, we call it minimum. Therefore, individual-based detection is more accurate estimation, allow more accurate estimation of infection rate. Therefore, we try to detect viruses from each individual mosquito. Objectives are to provide a more accurate estimate of dengue virus infection rate in mosquito by employing an individual-based detection method, and to assess spatial distribution of the dengue serotypes distribution in mosquito and patient during the same dengue outbreak period. Study area was Tarlac City in the Philippines here, and in 2015, our mosquito sampling period was in the highest peak season of dengue cases in Tarlac City, September, October. 1 month only, we did intensive mosquito field collections from 11 barangays in Tarlac City. Then our sampling design was like this. House suspected to have dengue infected mosquito. 48 were selected based on the report from hospital and villages. Then immediately after the report from them, we went to houses of dengue patients and their neighbors and collected mosquitoes using these mosquito traps, electric traps. We installed 24 hours at household, collected many mosquitoes. However, Egypti were very rare. Curex were very abundant. Then after collection, we preserved the samples in RNA at the field. The female mosquitoes only were used for viral detection because mosquito bite, female mosquito only bite humans. They extracted DNA from single mosquito, and we use multiplex real-time artificial assay to simultaneously detect and determine types of dengue virus. And we also got DNA sequence of dengue virus. And in parallel with mosquito analysis, we also analyze sequence of dengue viruses from isolate from patients. Then compare these two data sets. And this is the result. 11 barangays, villages. The analyze of females number in total 359. And this is number of positive, dengue positive mosquitoes, females, 10. So 10 out of 359 female mosquitoes, less than 3% were found to be dengue positive. And 7 out of the 48 households had dengue positive mosquitoes. These two columns indicate dengue serotypes detected from mosquito and human at each barangay. Then red barangay show different serotypes between mosquito and human. Black show the same. So 3 barangays were different in serotypes. So we expect to see same serotypes, assuming that the viral circulation between mosquito and human are occurring within barangay spatial scale. However, results were not. So this result suggests the viral transmission between human and mosquito are occurring in different places. For example, human in daytime, their place they're working or studying us in the school. Therefore, their places are different from their household. This movement may be the reason of this different stereotype result. Also, this distribution of stereotypes. So, blue dengue 1 and dengue 2 show similar distribution between human and mosquito. However, dengue 4 are very different. Mosquito here and patient here. Therefore, human movement or mosquito movement show this interesting pattern of distribution. However, maybe mosquito is difficult to fly to this distance. Probably patient or human movement is a factor causing this long distance viral movement. In this summary, detection and typing of dengue bird in one mosquito was successfully conducted using multiplex real-time RT-PCR assay. The fact that some dengue birds were found in geographically diverse locations indicated that there is active movement of dengue virus involving the transfer of infected humans and or mosquitoes. But finally, let me advertise about the 2 opening PhD positions in my laboratory in the 3 research areas with full financial support. Research area 1 is bioinformatics and genomics. Keyword is machine learning, genomics, genome-based species boundary inference or immune selection, environmental DNA, and environmental genomics. And research area 2 is antibiotic resistance C in water environment. So we'll see dynamics, spatial movement of ARGs in the catchment, focusing on hotspots. They are hotspots like water treatment plant or animal vegetable farms, something like this. And research area 3 is vector biology and virology. Mosquito genome and viral genome will be analyzed to see mosquito tolerance to viral infection and or viral evolution using in vivo or in vitro approaches. The application deadline is 28th of February 2021 or until the positions are filled up. This position just opened 10 days ago, therefore not so urgent. However, probably if you're interested, maybe better to submit your application document in this year. However, of course, next year is also okay. I suggest submit the document in this year. The start date of the question is between April to end October 2021. However, later start will be negotiable. The maximum 4 years and 6 months of PhD study in Japan will be conducted. Then you can combine your home country-based study, for example, field work or collaborative work with your current research institution or university. That is also possible or welcome. Then please visit our website or Facebook page for the application information. You can find this laboratory website, Ehime and Watanabe. Then please visit our laboratory website. Okay, thank you very much.